everybody, and welcome to the fourth of this year's Oxford Botanic Garden and Arboretum Autumn Science Lecture Series, sponsored by Plants, People, Planet and the New Phytologist Foundation. This is the first time we've ventured into the world of the hybrid lecture, so I do hope that this in-person and online viewing works well for everybody who has registered. My name is Simon Hiscock and I'm the Director of Oxford Botanic Garden and Arboretum, and as usual it will be my pleasure to introduce this evening's speaker, but only after I've done a few necessary preliminaries. For those in the room, in the remote possibility of fire or any emergency, a member of staff will be around to show you the way out. For those online, if you have any technical issues, please use the chat function on YouTube and a member of the technical team will get back to you as soon as possible to try and remedy the situation. You can also use the chat function to submit any questions you want to put to the speaker, and you can do this at any time during the lecture and towards the close. And the questions will be included in the Q&A at the end of the lecture. The lecture is being recorded and everyone who has registered will receive access to the recording via the email with which they registered. Those of you who have attended the first three lectures will know that the theme of this year's lectures is paleobotany and plant evolution. And tonight it's my great pleasure to introduce the very eminent paleobotanist, Professor Jenny McElwain from Trinity College, Dublin, where she holds the 1711 Chair of Botany and is also Director of the Trinity College Botanic Garden. Jenny studied botany as an undergraduate at Trinity College, and that's where she developed her interest in fossil plants, especially what they can tell us about past environments and climate. This interest led her to a PhD in paleobotany at Royal Holloway, University of London, and after that a postdoc position and a Leverhulme postdoctoral fellowship at the University of Sheffield. Here she studied the impact of carbon dioxide on global warming by examining plant fossils collected in Greenland during the 1920s. She then became assistant curator of paleobotany at the Chicago Field Museum of Natural History and later associate curator of paleobotany there. During this period, 2000 to 2006, Jenny developed a pioneering technique for using stomatal density as a proxy for carbon dioxide partial pressure, thereby allowing her to estimate past levels of CO2 by counting stomata on fossil leaves. Jenny's textbook, The Evolution of Plants with Kathy Willis, first published in 2003, quickly became a fixture of most biology undergraduate reading lists. In 2008, Jenny moved back to Ireland to take up a faculty position at University College Dublin, and in 2017 moved down the road to take up a chair of botany at Trinity College. In 2017 she was admitted as a member of the Royal Irish Academy, and in 2025 she was elected a Fellow of the Royal Society. Jenny is currently on sabbatical here in Oxford as a visiting fellow at All Souls. Jenny, welcome, and we look forward very much to hearing your lecture this evening.
Thank you very much. Thanks for the kind introduction. So evening, everybody. It's so lovely to see so many people. So thank you for coming out, on a late, cold evening to hear about old plants. And thank you very much to Simon and to Lauren for the invitation to speak here today. Can you all hear me okay? Great, okay. So my title of my talk is Trees Through Time, and I kind of had a panic when I looked at what I said I was going to talk about, because I kind of went down a wormhole on what is a tree, and it's actually quite complicated defining a tree. So we were so familiar with these beautiful structures, but I think the main message I want to get across today is we should not be complacent, and we should not take trees for granted, because trees have limits. They have climate limits, and we can see it in the world today. If you go to Greenland, there are no trees. It's too cold. If you go to the desert, there are no trees because it's too hot and too dry. So really, with the climate change in the future, we have to ask the question: are we going to hit the limits of trees in other parts of the world? But I think the thing I want to start with, and this is a magnificent ash in Trinity College Botanic Garden, is what is a tree? So I got very excited today looking at this, and I found this wonderful note, a note on the definition of a tree. And I thought, oh, this is brilliant, this is just what I need. But then if you look at how they define tree, in the note we will aim to represent some new assertions of the definition. A leaf is of a degree of and I suddenly realized this is a tree as defined by computer scientists in graph theory. So it's not a good definition of a tree. And so I went to this paper, and this is tree definitions by the Forest Inventory of Europe, and it's actually very technical. So a tree is a woody, and woody, that means it's got this really strong compound called lignin. It's perennial, that means it doesn't die back after it's produced. Of a species typically forming a single self-supporting stem. So a tree usually has a stem, but of course some trees are multi-stemmed, especially when you coppice them. So we have to say typically or usually. It is self-supporting. What does that mean? Well, there are structures which are huge, but they cannot stand alone. They have to be in a group, they're non-self-supporting. And a tree has to have a definite crown. So here we see a clear trunk and the crown. So this is the definition of a tree. Some people define a tree as being at least over 3 meters. Other people define it as at least over 10 meters. Some people define a tree based on the diameter at chest height. So what about tree ferns? Well, technically, tree ferns are not trees because they are not woody. What about these unusual structures here? These are the flowers of the century plants that are produced, every 50 to 100 years, a century. And these are actually the flowers. Now, interestingly, the stalks are woody. So does that mean this plant is producing a tree flower? But this is a flower, and it dies back after reproduction, so it is not perennial, so it is not a tree. What about bamboo? We say bamboo is woody, but actually it's not woody. And it's a grass. It's not what we would typically associate with a tree. And actually, a bamboo stem alone doesn't always support itself. So for my definition of a tree, I'm going to be a little bit looser and not so strict, because this is a talk about the evolution of trees. So we have to be a little bit less strict. So the definition of trees I'm going to take is actually a functional one. So a tree, what is the purpose of a tree? Well, the main purpose of a tree is to beat your neighbors, to get bigger so that you can reproduce better. So you can spread your pollen and spores and seeds and make sure the next generation survives. So under that functional definition, then the oak tree on your left and the tree fern are both trees. And so I'm not going to be so strict on what was the first tree in the fossil record by having to have a woody structure or the compound lignin. So that's my definition of a tree. So what about a forest? Well, a forest is not just a group of trees, because this, to me, is not a forest. It is wood production, but it is not a thriving, dynamic ecosystem. So actually, just by planting trees, you do not necessarily get a forest. A forest takes a very long time to evolve the complex dynamics. So a forest is really the complexity, the dynamics between all of the organisms, but it's always dominated by trees. So really today, what I would like to do is take you on a journey over geological time, and I want to stop off along the last 400 million years and look at some of the evidence we have by studying fossils for the oldest trees, and what are the limits of trees. So can we study the climatic limits of trees in the past? What have they survived? How are they resilient? So I've organized the talk into past, present, and hopefully future. If I don't go on too long, I'll talk about the future. And to start with, I'm going to look at trees through time and look at some of the fossil evidence we have for trees in the geological past. It was very difficult choosing, so I wanted to tell you about all of them, 390 million years of them, but that's not possible in an hour. So we're going to start all the way back, 390 million years ago. This is almost at the beginning of when land plants first came onto the Earth, which was around 450 million years ago. And this is the time in the Middle Devonian that we have evidence for the oldest trees in Earth history. So to orientate you in time, this is 500 million years all the way up to the present day. And don't focus on the globes initially, if we look on the bottom. So we can divide plant evolution into three great evolutionary groups. Paleophytic, the old plants; Mesophytic, the middle plants, which is the age of dinosaurs; and Cenophytic is the young plants, which is really the plants that have formed most of the plants we're familiar with in our modern world. Grass, that many of you will be familiar with, you have lawns. Grass is actually evolutionary really young. And the earliest trees belong to these groups of plants that reproduce by spores. So nearly all of our trees today have seeds, they're seed plants, but the oldest trees on Earth actually reproduce by spores. So we're going to look at this in the late Devonian. The world, the vegetation is very much spore-dominated, but seed plants had originated, but they're still just very small. And the oldest plants are actually from Devon. And this was just discovered by a team in Cambridge and in Wales in the last few years, and published just this year or last year. So these trees are extremely unusual. They are not woody, but you can see they're a tree stature. They have trunks, they have crowns. The crowns don't have true leaves. So here we have plants that were producing a tree-like form, but they still had not yet evolved true leaves. So they were photosynthesizing by little branchlets at the end of long branches. And these fossils, fossils which were used to do this lovely artistic reconstruction here, were found in Devon. So they're the oldest trees in the world, 390 million years old. Now, the next oldest, or slightly younger, were 385 million years ago. And these were the oldest trees until they were just pipped by the Devon find. And we find those in New York, and they're called Gilboa trees. On the left is one of the fossil trunks. It's not working too well. The trunk on the left, and on the right is a reconstruction. And again, these trees have a look of a palm tree, but they're not palms. They're actually more closely related to ferns. And if you look at their trunks, they have these long structures which go up the whole length of the trees, and they're thought to be roots. So it could be that these trees were formed by a trunk of roots, like our modern tree ferns. And then in Ireland, about 360 million years ago, and I'm sure across much of Britain, where if we found fossils, there we're beginning to see trees which look much more familiar. And this tree is called Archaeopteris hibernica. And here's an example of a tree where if we did a cross-section of the tree, we would see rings, and you would actually see wood. So this is a tree with wood. It's got this incredible compound lignin that is really hard to break down. And it has big side branches, much more familiar with the form of a modern tree. But the leaves it reproduces by spores. So this is such an unusual plant. It's like a hybrid. It's got a trunk like a conifer, and it's got leaves like a fern, and it's an extinct group of plants. But this is one of the first trees, this is the first tree that had true leaves. So by 360 million years, we have complex trees, 1 meter in width at the trunk, up to 40 meters tall, with leaves that were spreading their spores all around the landscape. So now we're going to take a step forward in time to the Carboniferous, and we're going to look specifically at the middle Pennsylvanian. So this is between about 312 and 307 million years ago. And this was a time, the great coal forests. So all of the coal reserves all around the world were formed by basically the undecomposed wood and trunks of some of the earliest trees in the Carboniferous swamps. So in time now, we are here near this arrow just here. Oh, no, it's working now. It's working, yeah. I mustn't have been pressing it properly. So there, and now in the Carboniferous, this is still a time in Earth history when the vegetation predominantly reproduced with spores. So now we have true wood, true leaves, true seeds, but seed plants are still very uncommon. They're the rarities on Earth. And this is an interval that I have studied. Rather than trying to understand the evolution of trees and wood, my interest with my research group is looking at how trees or how plants in the past are sensors of the environment and the atmosphere they lived in. So I use the fossils from the Carboniferous to actually study what was the atmospheric composition 310 million years ago. How warm was the climate? How different was it to our climate of today? If we went back in time to the Carboniferous, you would see lots of different tree forms, but they're all becoming trees in a different way. So in a way, what you have is an arms race. Who's getting bigger the fastest, and who can outcompete their neighbor and get to the light, which is needed for photosynthesis. So this is a slide from one of the experts on Carboniferous fossils, which is Bill DeMichael in the Smithsonian. And there are all the different tree forms you find in the Carboniferous on top. And below are actual fossils, which are about a few millimeters thick, that have been peeled off a coal ball. And what I want to point out is if you look at the anatomy and a cross-section of these trees, they're all becoming trees in different ways. So on the left, we have an inner piece, which it looks like wood, just like the cross-section of wood you'd see in a conifer today. But the outer part is a thick, corky bark. In the middle here, this one, the trunk is made up of tiny little structures, and these are actually roots. So this was, even though it is an extinct fern now, it was just like the tree ferns we see today, like Dicksonia and Antarctica. And this one had a growth more like bamboo. So they're all working out how to become a tree in different ways and competing in these swamp environments. But my interest was really, how can we use these fossils, the leaves from these trees, to reconstruct atmospheric composition millions of years ago? And this is a bit of a tricky slide. This happened earlier. That's why it's animated. So I'm going to tell you a little bit about some of the research that I've done, and also my research group, on reconstructing carbon dioxide levels, atmospheric carbon dioxide levels, during this interval of the Carboniferous. So the method we use, Simon mentioned in his introduction, is very simple. There's a relationship between the number of stomata on a leaf surface and the CO2 concentration in the atmosphere. And basically, as CO2 concentration increases, the number of stomata decreases. And the reason for this is because plants they're ecosystem engineers. They're so attuned to their environment. So when there's a carbon-rich atmosphere, plants develop fewer stomata. They get the same amount of carbon for photosynthesis, but the benefit is they lose less water. So plants are constantly balancing this carbon trade for water loss. And because the fossils I work on can actually be peeled off the rock. So this is an example of a 200 million year old fossil. It's a ginkgo leaf from Yorkshire. And just below, hopefully it's not too dark, the glowing bright green, they are fossil stomata, which are about 186 million years old. So a lot of the research I've done is working out how can we characterize or quantify this relationship here so that I can count fossil stomata and work out how much carbon dioxide there was millions of years ago. Now, I've made it sound quite simple. It is a little bit more complex, but I'm not going to go into all the nitty-gritty equations and details. Ultimately, we're interested in what's the unknown CO2 in the middle of that big equation, and I use both observations of the number of stomata on fossil leaves, but I also use carbon isotopes. And the reason we can do this is because plants, a lot of fossils you're probably familiar with, they are turned to stone, so they're inorganic. The fossil plants I work with the original organics are preserved. So not only can you peel it off the rock, you can also, with a UV light, excite the original hundreds of million-year-old molecules and look at the chemistry and observe the leaf surface in really fine detail. How do we calibrate the response? So we can use herbarium samples. So the Victorians, the Edwardians, they collected samples of plants. They pressed them and kept them in herbaria, like in Oxford, like in Trinity. And we know the carbon dioxide those plants developed in. So by looking at historical collections of dried plants, we can calibrate the response of stomatal density to CO2 over time. Another way of calibrating it is to do experiments. So these are some of my lab group here posing in white coats in an experiment where we actually simulate the CO2 concentration from the geological past. And we can observe how does the plant respond in those conditions, and we can calibrate in that way. And as with all science, it's much better to have an understanding of mechanism and why it works or how it works. So some of the most elegant experiments are kind of illustrated by this little cartoon at the bottom. And I had colleagues in Sheffield who ran some experiments where they grew Arabidopsis plants, and they let the old leaves, they built a little mini carbon dioxide chamber around the old leaves, and they let the old leaves develop in high carbon dioxide. And then they looked at the new leaves, the little bright green leaves, to see how many stomata they had. And the new leaves showed unexpectedly low stomatal density. And what this showed is that the plant must be sensing the carbon dioxide in the atmosphere and communicating to the new leaf to develop the perfect number of stomata for that CO2 atmosphere. So this experiment showed there must be a sensor, and we know what that is now. It's actually, we have it in our tongue, so it's carbonic anhydrase. So when you take a fizzy drink, you know it's fizzy because you can taste carbon dioxide. So plants have the same enzyme in their stomata, and they can sense the carbon dioxide in the atmosphere. And then there must be a molecular signaling pathway between old leaf to new leaf. So the method I use on fossils is really well characterized, and we have a good understanding of how it works. I think the best way of explaining my method, though, is through cake. And in Trinity, in the Botany department, we have Botany bake-off. and it's the most extreme competition there is. There's nothing as extreme or as difficult. So this is my Botany bake-off cake, and it's meant to be, it's a coffee cake, it was very tasty, with chocolate icing. And these are the stomata of a ginkgo fossil leaf, and they glow bright green under fluorescent light. And these are the subsidiary cells around the stomata. Each cell has a little bright green papillae, and my lovely CO2 molecules here going into the stomata, and Malteser water molecules coming out. But basically, that's what I'm looking at. I'm looking at fossil stomata to reconstruct CO2 in the past. And here's an example of some of the fossils I work with. So this is a Carboniferous fossil, Medulosa, and here are the beautiful stomata, really clearly on this specimen. So what have we shown with this work? So this is a reconstruction of how carbon dioxide changed between 311 million years ago and 290 or so million years ago. And you can see that carbon dioxide levels are going down, coming up, going down, coming up, going down, coming up. So this is based on the stomatal method, but also based on another method, just to double and cross-check it. And what we're seeing here are glacial, interglacial cycles, just like the last 2 million years of Earth history. And we show that the CO2 low is the same as during the last 2 million years, about 180 parts per million CO2. And the highs are slightly higher. They're 600 parts per million CO2. So with this work, we've been able to kind of provide a context for those forests of the Carboniferous. When it's really cold, the swamp forests on the equator that built all that coal, they expanded and proliferated. And then as we came from a glacial into a warmer phase, those forests contracted, the wet forests, and dry forests took over. But you've got this waxing and waning over time. Here, there was an extinction event, which I'm not going to go into today. But as soon as you've got a climatic context, you can start to try and understand why is it that those species went extinct. What is it about the species that survived that makes them resilient? And are there any lessons we can take from the past for today or for making predictions for the future? Keep an eye on time. I think I've got through good 50 million years, so I'm doing quite well. Okay. So what we also have done with these tropical coal forests in the Carboniferous is we've tried to understand how they functioned compared to modern tropical rainforests. So today, tropical rainforests on the equator, like in the Amazon, they're dominated by flowering plants. But of course, flowering plants have not yet evolved at this time in Earth history. So what we did is we looked at, again, looking at the stomata. It's a bit of an obsession. On the left axis here is stomatal conductance. So basically, it's a measure of how fast can that plant move water out of the plant body. And we compared the two on the left, Lepidodendrales and Medulosas, are Carboniferous trees. And we compared them to modern angiosperm trees and modern gymnosperm trees. And it was a huge surprise because what we found is that the Carboniferous trees, which are considered more ancient, moved more water through the plant body than even modern-day tropical rainforests. So the significance of this is that in the geological past, even though these are ancient trees, they're not angiosperms. We think that angiosperms are the most evolved. Plants were basically putting rain in the rainforest. When it rained in the Carboniferous, a huge proportion of that rain was recirculated by these Carboniferous forests. So what the plants were doing was actually keeping those wet swamp forests wet. And then a lot of the work we've been doing since is trying to understand if climate changes and it alters the forest composition, how will that then feed back on the climate system? And what you see is that plants that don't lose a lot of water can make the climate drier. And if plants which do move a lot of water are resilient to climate change, then they will keep the climate wetter. And we've been able to do this by basically applying modern models to fossil observations of these ancient plants. So now I'm going to, we're stepping forward in time. I've taken a big jump. So now we're going to look at the Triassic-Jurassic boundary. And this was a boundary which was a mass extinction boundary. So it gave rise to dinosaurs and a lot of the vertebrate fossils that you would be familiar with and interested in. And I was really interested in this boundary because we know it was a climate change event, but we didn't really know too much about how plants responded, which plants survived, which plants went extinct, and we really didn't have a good idea of exactly how much the climate changed by. So these were my interests. So again, the red arrow, we're on the boundary of the Triassic and Jurassic. Now you have an Earth which is dominated by seed plants. Plants which reproduce by spores, the ferns and the lycophytes, they are still present, but they're not as dominant. The seed plants are the dominant portion of the forests. So to do this work, the fossil localities I was interested in all occur in Greenland. So this was an absolutely wonderful opportunity to basically go on 2 National Geographic expeditions to East Greenland. And the localities are just up here, off the ice sheet, but you only have a one-month window in the year to go to these localities. To give you a sense of what it's like, it's absolutely beautiful. This is bog cotton, so any boggy places in the UK, it's very similar. It's Arctic tundra, so it's much colder, but it's a treeless vegetation. There are woody plants. There are Arctic willows, Arctic birches, but they grow flat along the ground. They literally brush your ankles. We basically flew to a nearest air base, and there's no roads, so we helicoptered to our field localities. I don't have that many photos, but to give you a sense, it's absolutely beautiful. We camped by a river. Here's our field localities to give you a sense of scale. And you basically start at the bottom, digging fossils, and you move up through the cliff, with your hammer and chisel. And if there is a fossil plant present, it forms a line of weakness. So when you hit it with the hammer, it opens up and you see the fossil is revealed. So I knew this locality had fossil plants because my supervisor's supervisor, so Tom Harris, collected these localities in the 1920s. And he did a beautiful piece of work, 5 monographs where he described described all of the plants down to species level. My interest in these localities was to try and look at the ecology in much more detail and try and work out which plants survived and what traits did those plants have, and which plants went extinct and did they have any particular traits. The fossils are absolutely exquisite. We know they're local because we get fossils like fiddleheads of ferns, which cannot be transported long distances. We collected over 4,500 fossils, and we collected them using ecological methods. So we were able to reconstruct these ancient forests of the Triassic. And all the way through this work, we worked with a scientific illustrator, Marlene Hill Donnelly, at the Field Museum. Extremely skilled illustrator, and this is her palette of greens, because she asked me, when she was trying to reconstruct and bring those black fossils to life, she asked, what color were they? And this was a really powerful question. I actually wrote a whole book about it, trying to answer it. It seems like a simple question, but it's actually very difficult. But there is science behind it. I can't go into it today, but we could work out broadly, were they bluey greens, were they bright greens, were they dark greens? So Marlene worked side by side with us, bringing these lost forests and lost landscapes to life, and she would illustrate pencil drawing, and then she would, after she'd done a pencil drawing, she would actually 3D model, and she would use kind of engineering principles. She would score the veins into the leaf and suddenly give it life because it's got an I-beam, basically. And then she would watercolor the 3D reconstructed plants. And step by step, we reconstructed what we could of the plant parts. Here's an example of a ginkgo relative called Baira. And you can see this is a paper model, but it has movement because she scored the veins in, and suddenly it stands up, so she could model it in a life position. And then we would work with the sedimentologists. So the sedimentologists study how were those fossils preserved? What kind of environment was it? So she would work with the sedimentologist asking questions, well, what was the ecosystem like? Did the plants occur on a bank, or do they occur in an oxbow lake, or is it an open delta? And then Marlene would sketch on location to get a feel of the sound and the feel and the right climate. So we'd work with climate modelers to try and reconstruct this whole feeling so that both scientifically and artistically we could bring all of these Greenland fossils to life. I love the sketches. I just think they're so beautiful. So this was one of the first sketches. We thought one of the plants may be a vine, so she reconstructed the vine up here in the top left. Positioning information, we collected very carefully. So each paleobotanist would dig a pit of about 1 meter by 1 meter cubed. So we knew exactly where the fossils came from in the landscape. So we're able to actually reconstruct the ecological positions. And on the left is a sunburnt me, much younger, sitting at the locality in Greenland today. And you can see it's an Arctic landscape. It's very desolate, there's no trees. And on the right is Greenland 200 million years ago. And you can immediately see without any data, without a reconstruction of CO2 or a climate model, this is a much warmer, wetter climate 200 million years ago than the one I'm sat in on the left, even though it was surprisingly sunny. So this is the reconstruction that Marlene did. In terms of ecologically, it was a cross between New Zealand's agathis or kauri forest and Florida Everglades. So although not on a tropical latitude, it was a tropical habitat at higher latitude 200 million years ago. I can't go into all the details, but beautiful ferns and benetites and cycads and conifers, broadleaf conifers, dragonflies, insect feeding damage, ecological interactions of a bank, and we knew which plants were in the floodplain. So all of this was incorporated, all the science was incorporated into the reconstruction. And then because these fossils are so well preserved, here's an example of a ginkgo on the left, and these are the stomata of the ginkgo on the right. You could actually compare the chemistry of the fossils to a modern ginkgo, and this is work by Margaret Collinson. But you can see chemically, the fossils are almost unaltered. They have the same chemical fingerprint as modern plants. So we can use all of that chemical information as well to try and understand what was going on at the Triassic-Jurassic boundary 200 million years ago. These are, I know it looks like leaf litter that on the paths of Oxford today, but this is actually 200 million year old leaf litter. So on the days when it was really windy in East Greenland, we couldn't work because as you opened up the fossil, they would literally fly away. It's a fossil leaf. And you can see some of them are very transparent. You can see the veins. You can even see a little feeding damage here of an insect. And then if you use UV light, you can see even more structures and detail. So we reconstructed the CO2 changes globally associated with the Triassic-Jurassic boundary. So there's the Triassic-Jurassic boundary. And what you see is that carbon dioxide levels increase dramatically. So 5 times higher than they are today. And we had fossil beds which occurred during this high CO2 phase. And what you notice is that it's very poor in trees. And this is a feature of all of the extreme environmental events in Earth history. We see a loss of trees. Ferns proliferate. If there's a volcanic episode, ferns always proliferate. They're incredibly resilient. So here is a meadow. There's no grass at the time because grass hasn't evolved. So a fern meadow proliferates in the peak of high CO2 and high global warmth. And the trees, many of those tree species go extinct or go into refugias to different latitudes, and we lose them from Greenland. And then in the Jurassic, you can see that forests re-emerge. So what I was saying in this, from these studies, is that plants are incredibly resilient. This is an extreme global warming event in Earth history, and yet forests, complex forests, re-emerged. It did take a long time, though. It took a good few 100,000 years. And many of the trees were related to those that were present in the Jurassic. So the cause of the Triassic-Jurassic extinction event and the cause of that increase in carbon dioxide was not humans. Obviously, we weren't around then. It was a 2 million year volcanic episode. And this released huge quantities of carbon dioxide and sulfur dioxide and other horrible organohalogens and mercury into the atmosphere. So it really was an extreme environmental episode. This was the final reconstruction of the peak of the volcanic interval. And you can see we have these red skies. And this is because we have huge quantities of fossil charcoal at this time in Greenland, and that indicates a high prevalence of fossil fires, of wildfires that we see fossilized in the charcoal record. And also we know from other studies we've done doing experiments with sulfur dioxide that there was huge quantities of sulfur. So these red skies are both kind of wildfire and volcanic haze. On the left is the reconstruction, the scientific and artistic reconstruction of Greenland, and on the right is our experiment. So we grew analog plants from today, relatives of those that are in Greenland, and we grew them in high carbon dioxide and high sulfur dioxide and low oxygen conditions. Horrible, horrible mix, which would basically, the oxygen was so low it would kill us, and the sulfur dioxide was pretty toxic. And you can see what happens to the plants. The plants are just totally fine. They're thriving. Now, as soon as they're hit with high SO2, they don't like it. But the point of doing these experiments is to try and understand, under controlled conditions, how does a plant respond? Chemically, anatomically, morphologically. Once we could understand how they would respond in an experiment, we could then go to the fossil and interpret it much better. So in the low oxygen experiments, we would actually have to wear a breathing hood. On the left, that's me and a postdoc, completely staged, because that's tomatoes, you probably have noticed. Tomatoes were not present at the Triassic-Jurassic boundary. But we did do the experiments, we just didn't take photos for a photographer. But these were the actual experiments with my postdocs and postgrad team. And what we found was really extraordinary. A single plant species is so plastic, it can modify every part of it itself under different atmospheric conditions. And this is illustrated here. So this is a very familiar plant, I'm sure, to many. It's Ginkgo biloba. If you grow it in just ambient conditions, very happy leaf. If you grow it in high sulfur dioxide, this tiny, crumbly brown leaf. So the tree constantly tries to produce a new leaf, but they fail, but the plant doesn't die. But knowing this information, we were able to pinpoint exact times in Greenland when there must have been high sulfur dioxide concentration, because we're able to calculate how leaf area changes. And then these are some illustrations of the recovery flora after. So within a few 100,000 years, this is what Greenland looked like. And although it looks probably at first very similar to the forests of the Triassic, we've done a lot of measurements on these forests, and we know that functionally it was actually very different. And the amount of water released from this vegetation was more than halved. So this is, in this forest compared to the forests of the Jurassic. So it looks similar structurally, but functionally it was very different. I'm going to skip this because I've gone on a little bit too long. So I'm going to finish very briefly with the present and come up to the modern day. And here, I mean, this talk series is led by Botanic Gardens, and although it's a paleobotanical talk series, I want to stress the point that Botanic Gardens are in a perfect position to ask questions about how plants will respond to climate change. We've done a lot of research in different biomes in the world, so going to forests all over the world, from Fiji to Alaska to Arizona. Wonderful fieldwork where we brought lots of undergraduate volunteers with us who absolutely loved the experience. And we've measured how plants are changing, how they move water compared to the 1980s. So on the map on the left, these are all the different forests we've visited. So for different biomes of the world, Fiji is tropical forest, Alaska is boreal forest. And we measured for the same forests about 20 or 30 trees in each forest. And we measured water use efficiency. So basically this is a measure of how efficiently plant takes up carbon dioxide and exchanges water. And what we found in the light blue is historical data that we gathered from herbarium specimens. So they was collected in the 1980s. And the dark blue is modern. And without fail, all the forests of the world are showing an increase in water use efficiency. So even since the 1980s, they're adapting to climate change and they're becoming more water wise. Within this data set, we can actually select the tree species that are more water wise than their neighbors. And we could use that information to make predictions about how they're going to respond to future droughts and future climate change. But I think it's really important that we couple this kind of field ecology work with more experimental work. And this is where Botanic Gardens come in. Because Botanic Gardens all hold living plant collections which are provenanced, are collected from all over the world. So you have this beautiful experiment, plants that were collected in Florida, New Zealand, North America, Ireland, all growing in what we call a common garden. So if you can track all those plants of different provenances to ongoing climate change using long-term data sets, we've got this incredible resource where we can actually track responses year by year. So in Trinity, I set up this project called the Witness Tree Project. And what we're doing is we're trying to track how trees respond to increases in carbon dioxide. So the data set on the left is the most famous data set, the most famous environmental data set in the world. It's Keeling, who started measuring carbon dioxide every day, every minute since the 1950s. And you can see there's a huge increase in carbon dioxide. The orange is from Mauna Loa, Hawaii, and blue is the record from the west of Ireland. And on the right is another cake. That is actually a cake of that's Mauna Loa, Hawaii, and it was an undergraduate first-year cake during lockdown. We had to make CO2 more exciting, so we did a bake-off. And that's actually candy floss clouds. Isn't that incredible? So we know how much carbon dioxide is changing, and what we're doing with the Witness Tree Project, we selected 21 trees. They are from different parts of the world. Some are native. They're from different hardiness zones, so you would expect some are more adapted to the future conditions, others less adapted. And we're measuring two traits in every tree. The first one is we're measuring their water use efficiency and how they move water. And as an analogy, have a look at the terrarium on the bottom left here. You can see water is condensating on the glass. Actually, what you're seeing is the effects of the plants in that glass vessel releasing water. So we have a lot of instruments that allow us to measure that really precisely. And that's our measure of climate resilience. And the other thing we're measuring is atmospheric pollution. We're measuring particulate pollution that affects human lung health and brain health. They're called particulate matter 10 and particulate matter 2.5. And although our ambition is to run this project, we've run it for 5 years so far, and we want to run it for 30 years. I'd love to run it forever. We need long-term data sets. And what's really extraordinary, even to date, we see that different species in our small botanic garden in Trinity have completely different capacity to remove particulate pollution from the urban environment. Indian horse chestnut, strawberry tree are really performing brilliantly, whereas the common street tree in Dublin, lime, is only capturing half of the particulate pollution. So what we want to do every year is monitor particulate pollution, how plants clean the air, and how they're resilient to climate change. And with that type of evidence, we can then make projections. I think you should select this tree for street trees in Dublin. I think this tree will be more resilient to droughts in the future. And that's the data on particulate pollution. So I'm not doing the future because I've already done too much. I'm going to go to the summary. So in summary, I hope I've shown that trees and tree-like plants have been on Earth for 390 million years. Trees from the past are witnesses to climate change. This is why we have a witness tree project today in Trinity, and we can use that data to try and understand fossils in the past, and we can use the data to try and project forward what's going to happen. The most important thing I want you to go away with from the lecture is that trees have limits. They have climatic limits, and paleobotany can help us understand what those limits are, but so can research in botanic gardens and in the field. Trees put the rain in the rainforest, and we've actually got ways of measuring how much different trees can recirculate water and make climates drier or wetter. Certain species of trees are resilient to climate change and atmospheric pollution, and we need the data to make good predictions of the future and to give good evidence-based, to develop good evidence-based policy. And I really think long-term monitoring is needed. Projects like the Witness Tree and other ongoing projects in botanic gardens all over the world. So thank you very much for your attention.
Thank you very much, Jenny. That was a very inspiring talk and plenty of ideas for new projects in botanic gardens as well. Yeah. So are there questions from the audience, first of all? Gentleman there.
So Jenny, back as a child in the 70s, we were misled to think this country had actually quite good weather. And then in 76, it was that very hot summer, and you could actually see the damage to the trees through that summer, that they were really impacted for the next 10, 15 years. And it's been interesting this summer that we've had the second really lovely summer of my life, and the trees have seemed to have survived. Are you suggesting that they sort of learnt their lesson back in the 70s? It's the same trees, presumably, who have done better? Yeah. Or how can a tree change to that degree?
Yeah, no, that's a really good question. So I suppose trees are really good to study this because they're long-lived. So we can, you could do a core and you could look at isotopes in every ring to look at how they responded. You could also look at their genetics, and even though it's the same genetics as this, epigenetics, which allows you to study how they're actually modifying their genetics as a living organism. But I don't think they are doing so well. So there's a lot of trees which are really suffering, and it's really hard to kill a tree. Like if I could do a lecture, or I couldn't actually, I'd have to really read a lot first. When a tree is dead, it's actually very difficult because it can lose whole branches and part of it, but still be alive and still resprout. And I know from research that's happening at Kew at the moment, they've got a lot of trees which are really suffering. and are already thinking of planting different trees. And I know then you get compound effects where you've got, like, we have a big issue with ash dieback in Ireland, and even though that's pathogens, I haven't talked about that. So I don't know whether that answers your question. I mean, some tree, the ones I predict which are going to be quite resilient are actually evergreens, and in the fossil record, this is what we see, proliferation of evergreens, which are kind of a bit slower, and they've got a tolerator strategy. And after mass extinction events, they're the ones that flourish. And it's the more deciduous, the kind of live fast, die young kind of strategy. They're very good at growing, very good at competing, and they're fast, but that isn't so good in a drought situation. So I don't know whether that answers your question or not really.
Yeah, it does. It's just that back in the 70s, you could, as you drove around, you could see the damage. I mean,
Yeah.
the trees were clearly maimed
Yeah.
by 76, and I haven't felt it was to the same degree. The trees obviously had a tough summer,
Yeah.
but they haven't actually been so
As hard,
visibly
yeah.
impacted.
But I suppose this is why we need the data. So what we do with this project is we now have, we measure 21 trees every year in Trinity Botanic Gardens, and now we have it replicated in the National Botanic Gardens, so on the other side of Dublin. So we have two teams collecting data at the same time, exact same time for the same leaf of the same species. So we've got these really nicely matched data sets. And the point of getting long-term data is that if there is a really severe drought or a severe heat wave or a really wet year, we will see it in the data, and we'll be able to say, oh, actually, that's an extraordinary year. We can only say that because we have got 10 years previously where everything's really happy. So this long-term data set's really important for that, yeah. If we'd had trees back in the 70s, we'd be able to say 70s was extraordinary, but we don't, yeah.
Thank you. Sue?
Fantastic lecture. Thank you so much.
Thank you.
I was interested in your comment on arbutus, which you said is really good at removing particulate matter. What is it about the arbutus that's driven that ability? Is it because it's a Mediterranean plant growing near volcanoes, or why is the arbutus so resilient?
And that's what we're trying, that's so interesting. So what we're doing is we're looking at all the traits. So we're looking at the leaf structure, microsurface roughness. Does it have a different cuticle? Do they have different hairs? One trait that seems to be very interesting is leaf angle. And we're double checking the data set now, but we think that's really important because it affects how wind and eddies move around the tree. And if it moves, and we're trying to model how the air is moving to see will the particulate load stick onto the leaf surface. And as soon as it's on the leaf surface, humans are out of danger, because then it rains and it washes into the soil, and it isn't a problem in the soil, it's only a problem when airborne. But yeah, that's really interesting. What you can do with this data set is ask that question. So we don't know yet, but that's what we are trying to work it out, yeah.
Do we have any questions from the online audience yet?
Not at the moment. We've got a lovely one here, if I may.
Okay.
Online audience as well. Put your person in. Do you have any evidence where you might find even older trees? Do you think you could find even older fossils?
Older fossils. Paleobotanists love finding the oldest and the newest and the new species. So I think there will be older ones out there. So what you do as a paleobotanist, you would look at old records, maybe old geological records, and the geologist wrote a note. You know, they're trying to understand what age were the rocks, and they'll write a note, twigs observed, and they'll draw them into or leaves observed, or maybe plant structures, plant roots. So you would try and find some of those geological notes to target localities where you know the rocks of the right age. Somebody previously, historically, has seen some roots. That's what you would target. So there will be paleobotanists trying to beat that 390 million year. I guarantee you they're probably somewhere in the world looking now in a different time zone. Yeah.
Okay, thank you.
There's a question here.
So given that obviously these forests over deep time are sort of being significantly influenced by how the climate is changing due to abiotic factors, but are also changing those abiotic factors, how, when both things are changing at the same time, how do you get a sense of like whether what the directionality of that changes, so whether plants are evolving to changes in climate or whether the climate is evolving to changes in plants? And does your trait-based approach in particular sort of allow you to tell the difference between those two directions?
Yeah, so that's a great question, the kind of chicken and the egg. Really the only way, one of the most powerful ways you can do that is to work with computer modelers. So like in my research group, I've got climate modelers, and what you would do is you would do a model experiment, and you would ask the question, if you change the vegetation, by how much does it change climate? If you change the climate, by how much does it change the vegetation? And then you try and look at these kind of model scenarios. In the fossil record, we haven't got the resolution, the time resolution to answer chicken and egg questions. You can hardly do it in the last 2 million years. You can't do it 200 million years ago. So this is where you have to work with modelers and try and kind of model some scenarios, and based on that, try and work out which came first. Yeah.
There's a question right at the back there.
Hi there. You were talking about how trees change with the changing climate. I mean, at the moment, the human-driven climate change is happening extremely rapidly. So there are trees in their native ranges which are now struggling, and trees in other places which weren't suited but are now very suited, you know, non-native trees. And there seems to be an obsession recently with restoring ancient forests in the UK, to the extent that they're cutting down established conifer plantations that have been left beyond their rotation age and are becoming quite interesting in terms of biodiversity. And yet we're seeing conifers in the Pacific Northwest which are now beginning to struggle. And the other week I was in the New Forest, and I was seeing coast redwoods regenerating in a way that they haven't done up until the last few years. Yeah. So what's your opinion on the idea of restoring something back to an ancient state as opposed to going with the changing climate?
Yeah, that's a great question, and actually I have a slide to answer, because the bit I missed, I don't know whether you can see that, but when I was going to talk about the future, we can actually look at the past. So this is taxodium swamps, so swamp cypress, Taxodium disticum that you find in Alabama and the bayous and the wet climates. So we have evidence for this across the Isle of Wight, Lough Neagh, Northern Ireland, 30 million years ago. So my own opinion is we have to be open to non-native as well. And because native plants in Ireland are defined in the last 10,000 years. So if it was present continuously, it is native. But we can't be so pure that we're closed to the idea of non-native, because if our climate, it is changing very rapidly. And it's a really difficult, I'd love taxodium street trees, or metasequoia, and actually I had another example, which was, so that's taxodium, but this is umbrella pine. So it's now endemic in Japan, but it was present in Galway, the west of Ireland, about 2 million years ago. And Galway could not support umbrella pine very well. It actually could grow it today, and it would certainly more in the future. So I think, obviously native plants are much better for hosting native biodiversity, and they host a huge biodiversity. But for some, perhaps, urban engineering, green engineering, we have to consider non-native trees as well. Yeah.
Plus, it might be that a mixture of native and non-native may be good in terms of not being able to spread in a
We'll have a question from someone in the online audience now. Lauren, fire away.
The question is, you said that the sensor of CO2 is carbonic anhydrase. Do you know from genomic data when that sensor evolved in plants? Is it present in the earliest terrestrial plants?
Fabulous question, and I can't answer it. But I do know that there's some really nice genetic work with mice and plants where they swapped the carbonic anhydrase in both and got functionality in the mouse with a plant-based carbonic anhydrase and vice versa. But when it evolved, if it's in animals and plants, it has to be really ancient. It has to go way back, yeah. So I'd say before both.
One more
but I don't know the
online, and
answer,
then we'll have a final one from the audience.
The other one I have online is, well, actually, I say it's online, it's me. I really want to ask about the ginkgo tree. I thought ginkgos were incredibly tolerant of pollution, and your data set had them at the bottom. So
disappointed with that.
Yeah.
Ginkgo, I thought it was, I won't go back, but my favourite plant, survived, 3 mass extinctions. And
they survived, wasn't it the 8 trees that survived Hiroshima as well, the ginkgos?
Yeah. So, but that could be why. A plant that captures a lot of pollution is good for us, but it might not be good for the plant. So ginkgo, we know, is a very good street tree, an urban tree. Maybe it can get rid of that pollution quicker. It's, the self-cleaning property of the leaves. But that was a real surprise to me as well, because I thought it would be trapping it.
I didn't jump the queue there, Simon. There wasn't another online question.
There's a lady here who's been putting her hand up quite a bit, so we'll give her the chance.
Hi, thank you. My question's about the preservation of the leaves in your leaf fossils.
Yeah.
You say you're able to excite the particles and do the isotopic analysis on them. Is it that the leaves aren't fully fossilized or mineralized within the rock, or is that something else?
Yeah, no, so the leaf, the inside tissue is called coalified, so it's turned to coal, but it's still organic material. So if you want to look at isotopes, you can scrape a little bit of that coal and look at the stable carbon isotopes. The cuticle is unaltered. So the upper and lower cuticle, and when you hit it with a UV light, it will excite the cuticle, and that gives off an autofluorescence. So you've got different sandwiches preserved in different ways, but if you've got a black fossil, which is coalified in the middle, then it most likely will have the original unaltered cuticle, which is incredible. Yeah.
Wow, thank you. That's very interesting.
Right, well, we'll pull it to a close now, and thank you again, Jenny. And thank you to the audience who have turned out on this very cold evening. And thank you all those sitting at home in the warm, watching online. So 2 weeks' time, we'll have the final lecture, and it will all be back online again when we're all covered in snow. So good night, everyone, and get home safely.